Alright guys, welcome to your 46th biology video, and on this video I want to touch real quick on a subject called leading and lagging strands of DNA. Now actually this subject can take like 5 different videos, but I just want to give you guys a real basic overview just because if I skip over it, people are probably gonna, get mad that I didn't touch on it. So let me go ahead and give you guys a real skinny on this. So basically, whenever your original DNA or your parent strand unzips, the two strands aren't identical to each other at first. They will be in the future, but at first, let's go ahead and take a note of what's going on. Remember that we said, according to the base pairing rules, that A always pairs with T and C always pairs with G. Well, this would mean as soon as they split that we would have our A and our C over here and our T and our G over here. So basically, as soon as this original parent strand splits, we have two little strands that are complementary to each other. Or sometimes I call them opposite, but technically they're called complementary. So even though what's going to end up happening is this T is going to go up here with a G, and we already know that T is always going to pair with a A, and G is always going to pair with a C, so this strand right here is going to be just like the parent. And also, remember this A and C over here? Well, this A and C over here are going to get paired with another T and another G, so you're like, you know what? Even though it splits, and they're not identical at first, you're going to end up with two strands of DNA identical to the original strand. Easy enough. So what's really the problem here? Well, for me to teach it like this, it's very easy, but for DNA polymerase, it isn't that easy because, and again, this is where I want to mention, forewarning, I could teach DNA polymerase, just this enzyme, for like 5 different tutorials and probably not have everything covered. So I just want to give you guys a real quick overview of whenever DNA polymerase, which was, the enzyme that's responsible for laying down that permanent DNA, whenever it lines up the nucleotides to make permanent DNA, it just can't line them up in any old-fashioned. It needs to line up the nucleotides in a very specific and certain way. Now, how this works, again, is beyond the scope. I suggest you read up on DNA polymerase if you want to get more involved. But just remember the overview of DNA polymerase needs to line up the nucleotides in a certain way to make DNA. So that's why you have one side that looks different than the other side, because the two strands aren't identical, and this can create some problems for DNA polymerase. So whenever your original parent strand unzips, it's going to split in two strands. Obviously, we know that already. Now the piece that's going to be one continuous piece right here is going to be called the leading strand. So Leading. Now, if you were to look at DNA under a microscope, you're going to see that one side of the DNA is going to grow in a continuous strand. This is called the leading strand, and another side of the DNA is going to be called the lagging, because unlike the leading strand, this is actually going to grow in chunks or fragments. So again, the side that is one continuous strand is called leading. The side that grows in chunks or fragments is called lagging. Now, basically what happens is once the DNA polymerase is done making these fragments, the DNA ligase right over here, that's where I said that whenever you have breaks, it comes and puts them together with covalent bonds. Well, that's what it does. It pretty much takes all those chunks and bonds them together. Now, I know this is kind of confusing because I drew the DNA ligase over here, and this is the leading strand, but I just drew over there as a demonstration because I was running out of room on the right-hand side. But actually, this DNA ligase enzyme would be over here, if this were a proper diagram. And what it would do is it would be putting together these pieces or these chunks using covalent bonds. So one last time, what happens is whenever your original parent strand of DNA splits, you have two strands that are complementary. One is the leading strand that grows in one continuous piece, and the other is the lagging strand, and this grows in chunks or fragments. And whenever we have our fragments, the DNA ligase is going to come with covalent bonds and put them together. So I know this can get kind of confusing, and DNA replication is actually pretty confusing, but pat yourself on the back because we're done with it for now. We're going to move on to something, a little more easier to understand. So thank you guys for watching, and by the way, before I let you go, I want to mention that if you don't really understand DNA polymerase and exactly what I talked about in this tutorial, don't really worry about it because you can understand the rest of cell division perfectly without it. This gets into the more advanced stuff. Really didn't need to make this tutorial, but now you have extra knowledge. Congratulations. So anyways, I'm rambling. I'm going to go grab some Hot Pockets. I'll see you guys in the next video.